Hi, my name is Tyler Davies, the research educator here at Cabrillo Marine Aquarium. I would like to welcome you all to the 2024 Young Scientists Symposium, where we'll be celebrating all of the hard work and dedication of our young scientists. Over the last 10 months, our students have proposed, designed, and conducted their own marine science research projects here at the aquarium. We hope you have a wonderful time celebrating these amazing students, and we hope that you learned something new along the way.
My name is Wesley Chung. I'm in 9th grade and I'm currently enrolled at Rolling Hills Prep. The topic of my YSP project is to figure out what variables triggered the small shark resting behavior where they pile on each other while resting. And I settled on this project because I noticed the small sharks in the exhibit hall, both pups and adults exhibit this behavior, and I want to know what variables trigger it. The success was that I figured out from the variables I chose which ones triggered the piling behavior and which ones didn't. And the knowledge I gained was that small sharks don't react to white, red, or blue light, but they swim away from green light. And they consistently piled when they had solid objects to rest on, underneath, or between, such as between the walls of the tank and the rocks that I had set up. Some advice I'd give would be to walk in with an open mind, and if they see an animal they are interested in learning about, they ask as many questions as possible about it.
My name is Inu Cho. I'm in 10th grade and I currently go to Palos Verdes Peninsula High School. My project is on the effects of adding a magnetic field into Chlorella vulgaris, which is a microalgae which is growing in popularity because it's so versatile, and apply it into wastewater treatment because I knew that wastewater and specifically pollution with nitrogen was a very prominent issue, and so I wanted to use Chlorella to remediate it. I think my favorite aspect of the YSP program was interacting with all the different young scientists and the mentors here. While experimenting in the amount of time that I spent here, I was able to have a lot of conversations with other people on what their projects are on. And then with the mentors, I was able to talk to them about what they're doing in their life, how their college research is going, what they're doing right now, and I was able to learn a lot from that. I think my biggest piece of advice would be to not be afraid to find errors in what you're doing, because for me, there was a lot that I was doing wrong, and I had to go through it all and solve all those different little details that I didn't really notice in my background research and in my proposal. And so that's what science is finding all these different errors and then learning how to solve them and overcome them, and then that's what goes into the final product, and it becomes your science project in the end.
Hello, my name is Kaylana Peng. I am in 12th grade and I go to Temple City High School. So my project is using algae in application for aquaculture, and I came upon this topic because I was considering working with algae in terms of upwelling, and so in terms of algae blooms and seeing what effects they have on the ocean in terms of that. But I was also interested in aquaculture and seeing how we could raise fish better for consumption, for general consumption. And so I decided to combine these two things, and I came upon green water aquaculture, which is basically using algae to help clean up the water in aquaculture tanks to create, to make it easier for the water to be filtered. So for my experiment, I ended up having a no results so far based upon my statistics and what I've seen in my data. And so part of the reason why this happened is because I was scared of using concentrations of algae that was too high, and so I ended up having algae concentrations that weren't high enough, and so my results were not very statistically different from each other. And so in the end, this is actually helpful, however, because we have created a baseline for where we can start to do more measurements and more experimentation and figure out what level of algae concentration can actually affect how much nitrogen is taken out of the water. So for the YSP program, first of all, I'm going to mention marine biology, so I've loved just being here in the aquarium at all and seeing the fish and the algae and just watching like our giant baby sea bass growing over the months that I've been here. Something else that I've really loved about this is how collaborative of an experience it is. I've been able to talk to the staff about my experiment and as well as other YSP students, and it's just been a very fun experience overall, and I feel very connected to this program, so that has been a good experience. First of all, don't be scared of your mentors or the staff. They are your best friends. They will help you for your project, throughout your project. I would say stay in very close communication with them. Let them know if you need any help, any resources at all. They will be very happy and glad to provide you with these. I would also say for the program, try and communicate again with your staff and your mentor in order to create a very clear idea of what you want to do for your experiment. This will help you set up your trials earlier, and don't be afraid to start a trial. Try doing mock trials earlier than you think you're ready. I think I was very hesitant to start a trial, and this resulted in my trials being pushed back a little further than I wanted them to be. And so don't be afraid to start quickly or start trying to arrange your experiment early and be ready for changes that may come from that.
Hi, my name is Ryan Kyanan, and I'm a junior attending San Pedro High School. The topic of my YSP project is about diatoms, specifically how do varying colors of light affect its growth. And what inspired me to choose this topic was actually here at the aquarium. There are white abalone that rely on diatoms as a food source, and I thought about delving into that topic to hopefully help them repopulate as they do need more food. I would say the most difficult part of my experiment was really deciphering what was diatom growth and what was other forms of algae like hairy algae. They do tend to be somewhat similar in color. However, hairy algae can be siphoned out, which is what I did to resolve that issue. From my experiment, we tested the effects of red LED lights, blue LED lights, and white LED lights. And as a result, the white LED lights showed the most effective growth and the fastest growth. The red was a close second. It covered about 80%, and the blue was the least effective in diatom growth. I would say the biggest piece of advice that I would give is to definitely have an open mind. Throughout your experiment, some things may not work out as you planned, so you'll just need to work through those situations and learn to adapt, and you can actually learn from those mistakes. And that could even be applied to how you even pick your research topic. The aquarium offers so many different things to research on, so you don't even know how fascinating something can be.
So my name is Mark Lin. I'm at Palos Verdes High School, and I'm in 12th grade, so I'm a senior now. My research project is based on an experimental medium that I invented last year, and basically how to take the algae out of that and use it for applications in both the biofuels and aerospace industries. I think the most difficult part about my research was designing a lot of new kind of methods. Last year I invented a new way to grow algae, and this year I have to invent a new method of isolating the algae. So I basically had to keep inventing and tinkering with different kinds of materials to try and come up to a good solution. And I'm just grateful because the YSP was able to provide me a lot of the resources that I needed to solve those problems effectively. There are so many things that are just awesome about YSP. I think the coolest thing is really the location and all of the people that come in. While you're working at the aquarium, there are a ton of different professors, researchers. I remember I met a PhD who was working with sustainable fuels and batteries, and we just talked for a really long time about how algae could be used in the industry and the current advancements in that area. I also once presented to a college class that just happened to be walking by. So, YSP just gives you so many opportunities to meet so many people, kind of advance your craft, and continue just loving science and scientific inquiry. I think the greatest advice I could give to aspiring YSP students is to reach out and don't be afraid to ask questions. Very often there's a lot of things in research that are just, we don't know. Whether it be, if you're a new student, like things like statistical analysis or how to grow algae a certain way or things like that. When you reach out and the staff here are more than willing to give answers, you end up learning a lot more, and that's what makes the YSP experience truly valuable.
Congratulations to all our young scientists on an amazing year of scientific exploration and achievements, from studies on microalgae to microplastics, abalone, sharks, extensive field and laboratory studies, artwork. You've all made incredible strides in the fields of environmental and marine science. We're so thankful and proud of all your hard work over the last 10 months, and we're so thankful to our friends group and all of our program sponsors. We couldn't have done this program without you. And to all of our young scientists and your families and your friends for your enthusiasm and dedication and support throughout this journey. Here's to another year of curiosity and discovery and making big waves in the world of science. So many congrats. Thank you, thank you, thank you. It's been an amazing year, and we're so excited to see what you do in your very bright future. Thanks.